So I'm going to talk today about work I've been doing at Sanger on looking at improving bird genome assemblies, and in particular problems that we have with assembling microchromosomes in birds. So bird genomes have an interesting organization. They're split into macrochromosomes, which are very large chromosomes. So here on the left-hand side, you can see a picture of the chicken karyotype. And we have these very large chromosomes. And then there's also a lot of tiny chromosomes in bird genomes. And most bird genomes have around 10 of these large chromosomes and around 29, 30 tiny microchromosomes. And these microchromosomes can be further subdivided into microchromosomes and dot chromosomes. And these dot chromosomes, of which there are typically around 10, are generally sized between about 5 megabases and 1 megabase in size. So they're quite small compared to these big sort of 60, 100 megabase chromosomes, which are the macrochromosomes. And they have interesting genetic and epigenetic properties. So they are hyperacetylated. They're very gene rich. So this picture here shows DAPI staining of chicken chromosomes and then fish staining with a CpG island probe, which is associated with genes. And so you can see that lots of these small chromosomes have a high density of staining, indicating that they're very gene rich. And so, for example, and this is supported by genomics as well. So in the cuckoo genome, for example, the 10 smallest chromosomes account for only 2% of the genome assembly content, but they contain around 6% of the protein coding genes in the assembly. So they're enriched for protein coding genes. And they also have this unusual organization in the nucleus as well. So microchromosomes tend to cluster in the center of the nucleus. This is done here with Hi-C modeling, with the microchromosomes predicted to be present in the center of the nucleus and the macrochromosomes around the edge of the nucleus. And this has also been shown experimentally as well. And interestingly, in falcons, which have chromosome fusions between dot chromosomes and large chromosomes, even when the microchromosome is fused to a larger chromosome, it still clusters within the center of the nucleus, indicating that it's actually either the sequence composition or some epigenetic characteristics of the chromosome which cause this clustering rather than just the size on its own. But in terms of genome assembly, what we find is that even though they only account for a tiny percentage of the genome assembly, they actually account for nearly all of the assembly effort when you're trying to both assemble and curate these genomes. They're incredibly challenging. So this is a typical Hi-C map of a draft bird genome assembly. So you can see here the large macrochromosomes in the assembly, this is just the draft scaffold. So they've been assembled with Hi-Fi-SM using Hi-Fi data and also with Hi-C data for scaffolding. And the large chromosomes come out pretty much whole out the box. And if you do a quick sort of pass through them, try and find all the chromosomes, in this goose, we can easily find 28 chromosomal elements and put those together. But we know from cytology that in this goose, the example of this goose, we actually expect 39 chromosomes plus Z and W. So that means that in the bottom of the assembly here, which has got all the repeat content that can't be placed, we'll actually have 13 chromosomes somewhere in this mess of repeats, which need to also be curated. And that's incredibly challenging. And so it's possible. So we use various sort of tips and tricks to try and find these tiny chromosomes. They tend to have this elevated Hi-C background due to their position in the nucleus, so we can use that to find them. And we use conserved gene mapping to try and pull out these scaffolds to build up chromosome models. So in this goose here, we did find all the chromosomes. And you can see the smallest chromosome here. This is a zoom in on the end of the assembly at these tiny chromosomes. You can see this high background Hi-C interactions. And the smallest chromosome here is around 150 kb in length. And these are typically in bird Hi-Fi genome assemblies, these chromosomes are very fragmented and made of very tiny scaffolds that we have to piece together, which is difficult. And as a result, when you look across all the released bird genome assemblies to date, so Sibele, who works at Sanger and is involved with the GOAT database, she put together a curated list of all the chromosome level genome assemblies of birds on NCBI that also have karyotype data, of which there are about 100 genome assemblies now with karyotype data. And if we plot out the assembly chromosome count versus the cytology chromosome count, we can see that over 50% of the genomes actually have fewer than expected chromosomes based on cytology, which reflects this difficulty in assembling these tiny chromosomes. And she actually made an interactive version of this figure. So if you scan the QR code here, you can view all this data in an interactive plot and see which assemblies are missing chromosomes. So if anybody wants to take a picture of that, feel free. She put a lot of effort into that. It's really good. Okay, and so really we wanted to try and improve these genome assemblies and figure out why they're so challenging and see if we can make them better. And recently there was a near T2T assembly released of the chicken, which included both hi-fi and nanopore data, and this seemed to fill a lot of the gaps in the assembly. So we were interested in using nanopore to try and assemble some of our bird species. So we instigated a pilot project at Sanger, where we generated Oxford Nanopore long-read data for 20 bird species from 9 different families. And all of these species we'd already generated bio-hi-fi and hi-C. And in 19 out of the 20 samples, we have the ONT, the hi-fi, and the hi-C from the same individual. So we can generate phased assemblies. And 17 of these species have already been released as hi-fi assemblies on NCBI through our pipeline. And we're going to use this to try and understand what's going on with these assemblies. And this is just a rough phylogeny of these 20 bird genomes plus the chicken T2T assembly as an outgroup. Well, not quite as an outgroup, but it's clustered down here. And this is just showing we cover quite a lot of bird diversity with these assemblies. Everything colored purple has got a released hi-fi assembly. And so far we've manually curated 4 of the nanopore assemblies as well, which are spread across this phylogeny. And due to time constraints, I'm mostly going to be talking about the herringull assembly BLAR ARG3 today, which is the one which we've done most of the analysis on, which is located up here in the phylogeny within Laridae. Okay. So Xenia in the Tree of Life assembly team had a look at the nanopore data that was produced. So part of the project was also optimizing nanopore data production at Sanger. We hadn't generated a huge amount of nanopore data in the past. And so we were looking at improving protocols for that. And so we were aiming to generate just basically completely standard nanopore. We weren't looking to generate ultra long reads, just the typical nanopore libraries. And we achieved between 5 and 15 kb read N50s, up to about 25 kb read N50s for some of the samples, and between 15 and 30x coverage per haplotype. So up to about 60x coverage in total with nanopore data. And we did find that we got much better results with the blood samples than other tissue samples. So that's something that seemed to work quite well when generating this data. And Xenia did a heck of a lot of work trying to optimize how to assemble this data well. So we wanted to maintain the ability to create 2 haplotypes rather than a merged assembly. And so we experimented with using hero read correction on the nanopore reads. We looked at using a program called RAFT, which breaks up long reads, which are the contained reads, which is a problem for hi-fi SM. And then recently they released a new version of hi-fi SM, which was able to natively assemble the R10 nanopore data without any extra read correction. We found that with hero correction, the assemblies all had quite a bit of KMA dropout when we looked at the KMA spectra. And we think this is probably something to do with hero correction that doesn't quite work ideally. The hi-fi SM with RAFT worked quite well. We were getting N50s around 12 to 15 megabases and reasonably complete assemblies. But we did find that this new version of hi-fi SM produces really good assemblies straight out of the box with the native R10 nanopore data. So we were getting 60 to 80 megabase scaffold N50s using the nanopore plus hi-C. This is without any of the hi-fi data. And we also got really good results with Virco, although we found that Virco didn't work well with hero-corrected data for these birds, but it did work well with the hi-fi SM corrected read. So that's like an extra step we had to put in to get Virco working. But for the rest of the talk, I'm focusing on these hi-fi SM ONT assemblies. Another thing to note is that when we use hi-fi SM with the hi-fi data, what I'm going to show later on is that the hi-fi reads suffer some sequencing dropout, which causes assembly problems. And if you still use the hi-fi data with hi-fi SM, as well as the nanopore as long reads, as ultra long reads, you still get sequence dropout in the assembly. And so we got the best results by just using the nanopore data on its own to generate these assemblies. And that was all done by Xenia. Okay, so what do these assemblies look like? So this is a curated assembly of Larus argentatus, the herringull. So here we can see we identified 34 autosomes plus 7W, which is the published karyotype for this species. It's a really nice assembly. We find lots of repeat content contigged into the chromosomes. And here at the end of the assembly, I've zoomed in on the 10 smallest chromosomes in the assembly. And this is fairly typical for well-assembled dot chromosomes, where you see these big clusters of satellite repeat, which is similar across most of the chromosomes. Makes them quite challenging to curate. And also these more euchromatic regions of the chromosomes. And these smallest chromosomes will range in size from 5 megabases down to 1.5 megabases. But if we compare this to the hi-fi assembly of the same species, the same sample of the same species, with that assembly, we couldn't find one of the autosomes in the hi-fi assembly. We only found 33 autosomes. And the dot chromosomes were considerably smaller. So the smallest chromosome in this hi-fi assembly was only 300 kb long. So the longest nanopore chromosome is substantially longer than the shortest hi-fi chromosome. There's a big difference in the size. We also saw similar results for other species. So this is the northern pintail. In the nanopore assembly, we found 39 autosomes plus 7W. And again, the smallest chromosome, when you zoom in on these dot chromosomes, was 1.1 megabases. Whereas in the hi-fi assembly, we only found 37 autosomes, so we had 2 missing chromosomes. And the smallest chromosome in this assembly was 108 kb. So the smallest chromosome is 10 times bigger in the nanopore assembly than it is in the hi-fi assembly. So we think we're probably missing quite a bit of content in these hi-fi assemblies, and we wanted to investigate that in more detail. So this is an alignment of the hi-fi assembly versus the nanopore assembly. And here you can see that we see good correspondence between the two assemblies. And then if we zoom in on the small chromosomes at the end of the assembly, so this is chromosomes 24 to 34 in this Larus argentatus assembly. And what you see when you look at the alignments of these small chromosomes is you see bits of the chromosomes from the hi-fi assembly aligning well, but then lots of shrapnel contigs, these unplaced scaffolds, also aligning to the nanopore chromosomes, and some big gaps in the chromosomes as well. And I think chromosome 27, for example, this is the missing chromosome in the hi-fi assembly. It was present in the shrapnel, but there's some missing content, and it's all just in these tiny unplaced scaffolds at the end of the hi-fi assembly. So yeah, it seems suboptimal for the hi-fi assembly. And so to dig into this in more detail, we had a pipeline to analyze these assemblies. And so we took the new nanopore curated genome assembly. I aligned all of the hi-fi reads, the nanopore reads, and we'd also generated RNA-seq data for all of these samples as well. So we could look at gene expression, align that data. I annotated it with an avian repeat library provided by Alex Su and Valentina Piona, which is like a pan-avian repeat library. I identified tandem repeats with TRF, and then I identified protein coding genes with breaker3 using protein alignments and the RNA-seq data. And then I also aligned to the nanopore assembly the curated hi-fi assembly to look at which regions of the hi-fi assembly are found in the nanopore assembly and which regions might be missing from the hi-fi assembly. Okay. So the first thing, just looking at the coverage data, so looking at hi-fi read coverage along nanopore assembled chromosomes here. So what we can see is that this was a very high coverage data set. So the hi-fi data is sequenced to about 210x for this goal. It's a really high coverage data set. And across the large chromosomes, the coverage looks really nice and consistent, around 200x for all of these sort of 12, 13 largest chromosomes. But then as we get down to the smaller chromosomes, the hi-fi coverage really drops off a cliff, and we get a lot of the chromosomes actually with very low, close to zero coverage. So there seems to be a problem with hi-fi sequencing these small chromosomes. And in the nanopore assembly, which we had sequenced to about 45x, we do see a drop-off in the smaller chromosomes, but only about 25% coverage drop-off, so much less drop-off. So it seems like the nanopore data is able to sequence through these chromosomes much better than the hi-fi data. And we can really dig down into this in a lot more detail. So this is a macro chromosome, and this is one chromosome, 60 megabases long. And I'm just going to talk you through the tracks. So at the top here, I've got alignment blocks of the hi-fi assembly aligned back to this nanopore assembly. Anything in coral is a chromosome in the hi-fi assembly. Anything in purple is unplaced shrapnel content from the hi-fi assembly. And then I've got hi-fi read coverage in gold, then nanopore read coverage in green, and then under that, we've got RNA-seq, satellite DNA, and transposable element density as well. So this is a large chromosome. It looks nice. The hi-fi coverage is a bit up and down, but generally we sequence through the chromosome really well. But if we go to one of the small chromosomes, this is one of the dot chromosomes, chromosome 34, and you can see straight away that the right-hand side of the chromosome is present in the hyphae assembly, and it's been identified as a chromosome. But the whole, about 1.5, sort of 75% of the start of the chromosome is either completely missing, that's in gray, or only in the shrapnel of the hyphae assembly. And then if we look at the read coverage, well, what we see is that through the bit that's in the hyphae assembly, it's actually quite low, but it does sequence through. But then in all this missing part of the assembly, the hyphae read coverage is super low and dropping close to 0 in a lot of places. And this is a big stretch of the chromosome. It's not just in small bits of the chromosome. It's over like a megabase scale region. And it's not just repeats. There's lots of gene expression in this region as well. So it's protein coding genes as well as repeats. Okay. And then we can actually look at this in a bit more detail, say in IGV. So here, this is a zoom in on chromosome 32. You can see very clearly this is a gene-dense region, small region with multiple protein-coding genes. And hyphae coverage in these regions is actually going down to 0 in a lot of cases over these protein-coding genes, whereas the nanopore is sequencing all the way through the assembly. So it seems like the hyphae is having a problem sequencing these gene-rich regions of microchromosomes. And this is just zoomed in a bit more. You can see that the end of this protein-coding gene has got 0 coverage with the hyphae data, but is covered perfectly well with nanopore. So to quantify this genome wide, we took all of these missing regions in the hyphae assembly, and then looked for protein coding genes and counted them in these regions. And so genome wide, the hyphae assembly is missing around nearly 400 genes. So a very large number of protein coding genes are missing from the hyphae assembly, which were assembled in the nanopore assembly. So I think this is quite a big problem. And if we look at the distribution of those missing genes, we can see they're all grouped around these small microchromosomes. And in total, we see this is shown here as a proportion of the genes per chromosome. So in these dot chromosomes, up to 65% of the protein coding genes are missing from the hyphae assembly. Okay. So we were interested in maybe what could cause this dropout. And so one thing we know, this is kind of very much work in progress, but one thing we have noticed is that there's seems to be interspersed with these gene-rich regions, we see a lot of satellite DNA. And we know that in fish, for example, PacBio hyphae suffers from GA-rich dropout, and maybe some of these satellites have a lot of GA repeats, but we haven't done an in-depth analysis as yet. But I just want to highlight that it's quite striking. We see, so here, this is chromosome 29. We see this region at the end, which has got a lot of gene expression, where the hyphae coverage drops away, and it doesn't get assembled in the hyphae assembly, but it does have this high density of satellite DNA. And we see this in other species as well. So this is the Anisacuta assembly, chromosome 38 in Anisacuta. And we see the first half of the chromosome is very repetitive. It's nearly all transposable elements, hardly any gene expression, very repeat rich. The second half of the chromosome is loads of gene expression. It doesn't get sequenced properly by hyphae. Nanopore is able to read through, but we also see just where the hyphae drops off, we see this increase in satellite DNA density. So it seems like maybe these gene-rich regions of the dot chromosomes in particular are interspersed with satellite repeats, which could be causing a problem with hyphae sequencing. Okay, so I'm just going to summarize that. So basically, we found that PacBio hyphae suffers from severe coverage dropout in bird dot chromosomes, and I think this is quite important because nearly all bird genomes, new bird genomes, are done with hyphae. So it's quite, and from looking at things that we curate, basically all the birds we see, the hyphae assemblies all look very similar in terms of very high fragmentation of dot chromosomes. And these dropout regions are in the worst possible places, so they're associated with protein coding genes. Up to 65% of genes are missing in the hyphae assemblies. So we see similar things from 3 different families of the 4 assemblies we've completed to date with nanopore. The good news is that just by using completely bog standard nanopore, you can recover these regions and assemble them properly, which is great. And we do find that combining hyphae with ONT does still lead to dropout. So we need a bit more looking into that in more detail to maybe work out ways to use the hyphae data that we have. We found that using this new hyphae SM version with the native ONT data base called with Dorado in super accurate mode gives the best assemblies, and it's also straightforward to use. but we also got good results with Virco. And we've potentially these hi-fi dropout regions are associated with tandem repeats, but we need to do a lot more work to confirm that. Obviously, there's a huge team at Sanger. Xenia did loads of work on optimizing the assembly strategy for the nanopore data. And I also just highlight that if you're struggling with hi-fi assemblies of birds in particular, I've developed a tool called Microfinder, which uses conserved gene content to pull out these fragments of dot chromosomes, move them to the beginning of the assembly, and help you curate the chromosomes more efficiently. And if you follow the QR code there, there's a tutorial on how to use it, a YouTube video, and a whole bunch of example assemblies which you can download to have a go with. Take any questions.